Hello guys, I am D.D. I am expert of botany. Today I will start respiration. What is respiration? Respiration is oxidative, amphibolic, exergonic, completely enzymatic controlled, biochemical reaction. Dear guys, it is the 5 terminology. From here question will be also asked in NEET examination. What's respiration is oxidative. What is meaning of oxidative? Oxidative means glucose molecule is oxidized into carbon dioxide, or you can say oxidation of glucose into carbon dioxide. That's why this process is known as oxidative. Okay? Are you getting my points? Thank you. Second step, respiration is amphibolic. What is meaning of amphibolic? Means anabolism plus catabolism both are combinedly known as amphibolic. Means in amphibolic process, anabolism and catabolism both process take place. Anabolism means synthesis of any molecule, and catabolism means destruction of any molecule, that is known as catabolic. So then respiration is amphibolic process. Why? Because glucose is oxidized and energy is produced, and from intermediate compounds like OAA means oxaloacetic acid and alpha ketoglutarate acid, synthesis of some amino acid also occurs. Third point, exergonic. Respiration is exergonic process. What is meaning of this? If energy is released from any reaction or any process, that is known as exergonic, or you can say exothermic. Means if chemical reaction occurs inside our body, it is known as biochemical reaction, and if energy is produced from biochemical reaction, it is known as exergonic. Then respiration produces energy, it is known as exergonic. Completely enzymatic controlled. Respiration is complicated process. It is not a single step process. Minimum more than 25 step process. That's why it is complicated process, and this process is completely controlled by enzymes. That's why it is known as enzymatically controlled process. Biochemical reaction, the reaction which occurs inside the living organism. Bio means life, life means living organism. If chemical reaction occurs in the living organism, it is known as biochemical reaction. Okay? Again, just you listen. Occurs in all living organisms. Either in the presence of oxygen or in the absence of oxygen. Okay, respiration occurs in all living organisms either in the presence of oxygen or in the absence of oxygen, in which complex organic food material are either completely oxidized or partially oxidized. Into simpler form. At last, energy is liberated. That is known as respiration. What I am saying, just respiration is oxidative, amphibolic, exergonic, completely enzymatic controlled biochemical reaction occurs in all living organisms either in the presence of oxygen or in the absence of oxygen, in which complex organic food material like carbohydrate, fat, protein, etc. are either completely oxidized or partially oxidized. At last, energy is liberated. It is known as respiration. That's all. But now I will discuss about the type of respiration. Respiration is of 4 types: aerobic respiration, anaerobic respiration, floating respiration, and protoplasmic respiration. So here I will discuss first about aerobic respiration. Aerobic respiration. Dear guys, aerobic respiration means this type of respiration occurs only in the presence of oxygen. In the presence of oxygen. Okay, and this aerobic respiration is completed into 5 steps. It is completed into 5 steps. Number one, glycolysis. Number 2, ODC. ODC means oxidative decarboxylation, or you can say link reaction. Third, Krebs cycle. Fourth, electron transport system means ETS. And fifth, oxidative phosphorylation. Okay, first glycolysis, second ODC, or you can say link reaction, third Krebs cycle, fourth ETS, and fifth, that is oxidative phosphorylation. Okay, but first I will start glycolysis. Okay, then first you understand what is glycolysis. Glycolysis means stepwise degradation of glucose molecule into pyruvic acid is known as glycolysis. Clear? And this glycolysis is 10-step process. It is not a single step process. Understood? And it occurs in the cytoplasm of all living organisms, either eukaryote or prokaryote. Clear? It means it occurs in the cytoplasm, and oxygen is not essential for this process. What is the meaning of this? It means glycolysis is totally anaerobic process. Okay, but it is common in aerobic and anaerobic both. Clear? Just you see glycolysis. Beta, I am going to start step of glycolysis. Glucose is converted into glucose 6-phosphate. Clear? It is the first reaction, first step. Glucose 6-phosphate is converted into fructose 6-phosphate. It is second step. Fructose 6-phosphate, it is converted into fructose 1,6-bisphosphate, and this fructose 1,6-bisphosphate, it is converted into dihydroxy acetone phosphate and 3-phosphoglyceraldehyde PGALD. It means phosphoglyceraldehyde, and DHAP means dihydroxy acetone phosphate. Beta, both are interconvertible. Both are interconvertible. Okay. At last, dihydroxy acetone phosphate, it is converted into 3-phosphoglyceraldehyde. It means here 2 molecule of 3-phosphoglyceraldehyde is formed. Clear? What I mean to say? Means from one glucose molecule, 2 molecule of 3-phosphoglyceraldehyde is produced. Okay? Again, this phosphoglyceraldehyde, it is converted into 1,3-bis PGALD, means phosphoglyceraldehyde. Okay? And just you see, this 1,3-bis phosphoglyceraldehyde is converted into, 1,3-bis PGA, 1,3-bis phosphoglyceric acid, or you can say phosphoglycerate. Again 1,3-bis PGA, it is converted into 3-PGA. 3-PGA means 3-phosphoglyceric acid, or you can say 3-phosphoglycerate. This 3-PGA is converted into 2-PGA. 2-PGA means 2-phosphoglyceric acid, or you can say 2-phosphoglycerate. 2-PGA is converted into 2-PEP. PEP means phosphoenol pyruvate or phosphoenol pyruvic acid, and 2-PEP is converted into 2-pyruvic acid. Clear? It's the total 10-step process. Step number 1, glucose is converted into glucose 6-phosphate. Okay? Please concentrate. And glucose 6-phosphate, second reaction, it is converted into fructose 6-phosphate. Third step, fructose 6-phosphate is converted into fructose 1,6-bisphosphate, and fructose 1,6-bisphosphate, it is 6-carbon compound. It is broken down into 3-carbon compound, dihydroxy acetone phosphate and 3-phosphoglyceraldehyde. Both are 3-carbon compound. Okay? And finally, this dihydroxy acetone phosphate is converted into PGALD. That's why 2 molecules of 3-phosphoglyceraldehyde are produced by 1 glucose molecule. Clear? This PGALD is converted into 1,3-bis PGALD. Okay? Means 1,3-bis phosphoglyceraldehyde. After that, 1,3-bis PGA. PGA means phosphoglyceric acid. What is meaning of this? Aldehyde is converted into PGA, means acid. Aldehyde is converted into acid. From 1,3-bis PGA, 3 PGA already produced. From this 2 PGA, then 2 PEP, PEP means phosphoenol pyruvate, then 2 pyruvic acid. Key product, final product of glycolysis is pyruvic acid. Okay, 2 PA means 2 molecule of pyruvic acid is formed by 1 glucose molecule. Here glucose molecule is 1. Okay, again just you see in step number 1 beta, what will happen? ATP, 1 ATP molecule is used up in step number 1. The ATP used up, it is converted into ADP plus IP. Okay, that's why 1 phosphate group already added in glucose and forms glucose 6-phosphate. This glucose 6-phosphate is converted into fructose 6-phosphate. Both are isomer. Again, fructose 6-phosphate is converted into fructose 1,6-bisphosphate. It means in third process also 1 ATP molecule is used up. ATP is used up in third step. After that, fructose 1,6-bisphosphate, it is converted into 2 molecules here, dihydroxyacetone phosphate and 3-PGALD. Okay. Next, 1,3-bis-PGALD means when aldehyde is converted into acid, then dehydrogenase enzyme already activates, and here NAD plus, beta NAD, it is cofactor, means factor. This NAD plus is reduced into NADH plus H plus. Clear? Again, 1,3-bis-PGA means 1,3-bis-phosphoglyceric acid means phosphate group here 2. Okay, so 1 phosphate group already broken, then it is converted to 3-PGA. So here ATP is produced. Remember it. ADP plus IP and ATP produced. Again 3-PGA, 2-PGA, 2-PGA to 2-PEP, and from 2-PEP to pyruvic acid. PEP means phosphoenol pyruvate. Phosphate group here, beta. In pyruvic acid, phosphate group is absent. It means breakdown of phosphate group here. Then ATP also produced. ADP plus IP, it is converted into ATP. Clear? Just see step 1, 2, 3, here step 4, step 5, step 6, step 7, step 8, step 9, step 10. Clear? Once you see, question will be asked what where ATP is used up, step number 1, step number 3. At 2 steps means step number 1 and step number 3, 1 molecule of ATP is used up. Okay, and from which step ATP is produced? Just you see step number 7 and step number 10. 1 molecule of ATP here produced. But just you see, here you have taken 1 glucose molecule, but here already 2 molecules of PGALD produced. It means all molecules 2 produced. All molecules 2 produced. Then we can say 2 molecule of PEP, 2 molecule of PA, all are produced. So here for ATP, 2 ATP produced and 2 ATP produced. But before this step, here only single step, then only single ATP used up. But from 4th step, just you see here, means NADH2 already produced. So here 2 NADH2 produced and 2 ATP are produced. Clear? Again just you see enzyme. First step is regulated by hexokinase. Hexokinase. in the presence of magnesium ion. But a magnesium ion is the cofactor. Okay, means magnesium ion already activate to this enzyme, then this first reaction already completes. After that, in second, glucose 6 phosphate is converted into fructose 6 phosphate. So here isomerase enzyme, both are isomer, then isomerase enzyme already activates here. After that, fructose 6 phosphate is converted into fructose 1,6 bisphosphate. It means here phosphofructokinase enzyme, phosphofructokinase enzyme activates. After that, if one big molecule is broken into two small molecule, then enzyme aldolase already activates. In the presence of aldolase enzyme, two molecules are produced. After that, 3-phosphoglyceraldehyde is converted into 1,3-bisphosphoglyceraldehyde. Means step number 5, dear guys, just listen. Step number 5 is non-enzymatic process. It is non-enzymatic process. Non-enzymatic process. Okay, and 1,3-bisphosphoglyceraldehyde is converted into 1,3-bisphosphoglyceric acid, means aldehyde into acid. So here dehydrogenase enzyme activates. Okay, and in 7th step, 1,3-bis-PGA to 3-PGA. What is the meaning of this? One phosphate group is broken. Then here enzyme transphosphorylase enzyme activates. Okay, 3-PGA is converted into 2-PGA. What is the meaning of this? 2-PGA. It means phosphate from third carbon is changed into second carbon. So here mutase enzyme already works. 2-PGA is converted into 2-PEP. Enolase enzyme already works. Here last product pyruvic acid, then we can say pyruvic acid kinase enzyme. Pyruvic acid kinase enzyme. This enzyme in the presence of magnesium ion. Okay, guys, just you see, enolase enzyme is activated in the presence of magnesium ion and manganese ion, and here dehydrogenase enzyme. Okay, here pyruvic acid kinase magnesium ion and potassium ion. All steps are completed. First, again you see, glucose, single glucose molecule participate in glycolysis. In the presence of hexokinase and magnesium ion, glucose 6-phosphate is already formed. One molecule of ATP is used up. Okay. In second step, glucose 6-phosphate is converted into fructose 6-phosphate in the presence of enzyme isomerase. Why? Because both are isomer. And fructose 6-phosphate is converted into fructose 1,6 bisphosphate. It is due to presence of ATP. Phosphofructokinase enzyme already works here. Clear? And fructose 1,6 bisphosphate is converted into dihydroxyacetone phosphate and 3-phosphoglyceraldehyde. 3-phosphoglyceraldehyde beta, it is converted into 1,3-bisphosphoglyceraldehyde in the presence of H3PO4. H3PO4, 2 molecule, then 2 molecule this, and here 2 molecule of H2O, water molecule also used up at step number 5. Okay, and step number 6, it is the 6. Here 2 molecule of H2O produced, And step number 9, just you see here also 2 molecule of H2O produced. Clear? It means it is totally, glycolysis totally finished. And which type of question already has been asked in NEET UG from glycolysis, and which type of question will be asked in future also? Just you see. First point, glycolysis means stepwise degradation from glucose to pyruvic acid is known as glycolysis. Key product means final product. Just you see here, it starts from glucose and end in pyruvic acid. It means key product of glycolysis is pyruvic acid. Okay, from one glucose molecule, how many pyruvic acid is produced? Tell me. 2 molecules. Understood? And next point, how many ATP molecules are used up during glycolysis? Then answer will be 2 molecules. How? First molecule of ATP used up at step number 1 and second ATP used up at step number 3. It means in whole process, in glycolysis process, total 2 molecule of ATP are used up, are consumed. Okay, it's the question of NEET. How many molecules of ATP are used up in glycolysis? So only 2. Okay, how many ATP produced in glycolysis? Just you see beta, ATP already 2 ATP produced here at step number 7 and 2 ATP produced here, that is the step number 10. The total 4 ATP are produced, but 2 ATP are used up. That's why net gain of ATP equal to 2 ATP. What I am saying, it is also NEET question, NEET UG. Many times question has been asked from this point. What? Net gain of ATP during glycolysis. Just you see, 2 ATP here, 2 ATP there, 4 ATP already produced, and 2 ATP, 1 here, 2 ATP used up. Then what is net gain of ATP? 4 ATP minus 2 ATP is equal to 2 ATP. The net gain of ATP is equal to 2 ATP. Many times question has been asked from this point in NEET UG. Okay. Next question, how many water molecules are produced? How many means total water molecule produced during this process. Just you see, step number 6, here 2 molecule of water produced. Step number 9, 2 water molecules are produced. Clear? It means total 4 water molecules are produced during glycolysis process. And how many water molecules used up? At step number 5, just you see here 2 H2O molecules are used up. It means 4 molecules of water already produced and 2 water molecules used up. Then net gain of water molecule, the net gain of water molecule is equal to 4 minus 2 equal to 2 H2O. It is the NEET point. Clear? Again, how many NADH plus H plus are produced? Just you see here, step number 6, 2 molecule of NADH produced, and it is produced in the presence of dehydrogenase enzyme. Okay. It's also NEET question, how many NADH2 molecules are produced during glycolysis? 2 NADH2. Or you can say 2 NADH plus H plus. Understood? Again, what type of question will be asked? But just you see here, I have told means ATP, H2O, NADH2. But here question will be asked, total energy produced during glycolysis. Then total energy 4 ATP plus 2 NADH2. Clear? And when NADH2 enters into ETS, then by 1 NADH2, 3 ATP produced, total 6 ATP, and here 4 ATP. It means total 10 ATP are produced. 10 ATP are produced. It is the total energy. Understood? But net energy produced during glycolysis, beta 2 ATP already used up, then minus 2 ATP, it is equal to 8 ATP. It means 8 ATP molecules are produced, that is the net energy of glycolysis. Understood? And when 8 ATP already produced, when mass subtle, MAS, mass means malate aspartate subtle, malate aspartate subtle, when this subtle activates, then total 10 ATP are produced and net energy 8 ATP are produced. Okay? Why? Because mass subtle, if mass subtle activates, then by 1 NADH2, 3 ATP are produced during ETS. Okay? If GPS subtle, GPS means glycerol phosphate subtle, glycerol phosphate subtle, when this subtle starts, it is slow subtle. Slow subtle. What is meaning of this? By 1 NADH2, only 2 ATP produced. Okay? It means total energy 8 ATP minus 2 ATP, that is 6 ATP. When glycerol phosphate subtle already activates, then 6 ATP already net gain of ATP produced during glycolysis. Clear? Again, beta, step number 1, step number 3, step number 10. All 3 steps are irreversible. Irreversible means A is converted into B, but B is not converted into A. That is the irreversible. Okay? So step number 1, step number 3, step number 10. All 3 steps are irreversible and remaining all steps are reversible. What is meaning of irreversible here in case of glycolysis? It is the checkpoint of glycolysis. Okay? But out of 3 reaction, step number 3, what? Step number 3, it is most important control point of glycolysis. Glycolysis means glycolysis can be checked here at step number 3, step number 1 and step number 10. Just you see here, beta, why it is most important control point of glycolysis? Due to presence of phosphofructokinase enzyme. Phosphofructokinase enzyme. This enzyme is known as allosteric enzyme. Allosteric enzyme. Okay? Allosteric enzyme means it is activated in the presence of ADP and AMP, not ATP, and it is deactivated in the presence of ATP. It is known as allosteric enzyme. That's why phosphofructokinase enzyme, it is known as pacemaker of glycolysis. Pacemaker of glycolysis. Understood? It's the whole soul glycolysis. Thank you very much.